Hello everyone, and welcome to our fourth lecture in Unit 1. In this lecture, we are going to go over some of the basic techniques we use in a microbiology lab setting to help us grow, visualize, and identify certain microbes. This lecture really is designed just to give you kind of some basic ideas, some basic vocabulary, basic concepts, etc. about these techniques that we use so that A, you know what I'm talking about in later units when I mention these types of things, and B, you're a little bit more prepared for some of the material that you'll be going over in lab. So as usual, now you have your lesson objectives. These are, of course, used to guide your reading, make sure you're understanding the major topics in the chapter and in the lecture. So you can look at them before you go over this lecture, and then you may want to review them again afterwards to make sure you've hit all of the main points. There are a lot of different techniques that are used in microbiology labs to grow microbes, to analyze them, to identify them. But we're going to go over some of kind of the broad concepts and big techniques that are used very commonly in all types of different labs. And just to give you an overview to kind of put this in perspective, when we're working with microbes in labs, typically our goal is to grow them, to isolate them, which means to get them by themselves, and to identify them or figure out what they are. The main methods we use for that are culturing, which is what we use, or the methods that we use to grow the microbes, staining, which provides color to the microbes because most microbes are clear, and if you try to look at them, you don't see much unless you provide them with some color. We use a microscope or some type of microscopy to look at the microbes or to visualize them. And then to identify them, there are actually lots of different techniques, but some of the main ones are looking at their biochemistry or their metabolism and then doing genetic analysis on them. As you know by now, or at least as I hope you know by now, microbes are incredibly small. And for us to be able to study them effectively, typically we need to have millions or sometimes billions of individual cells or individual microbes to actually be able to do anything with them. One of the ways that we get this high number of microbes that we need to be able to study is to grow them on some type of medium or media. Medium is the singular term for what I'm about to be talking about, and media is the plural term. So a medium is some type of substance that provides space and nutrients for microbes to grow. You are looking at several different types of media right here. Media can come in lots of different types. There is liquid media, there is solid media, there is what we call general purpose media, which has a lot of different nutrients in it and allows a lot of different types of microbes to grow. And there's also things like specialized medium, which you're looking at down here on the bottom part. And specialized or specific media has certain environmental conditions and provides very specific nutrients that allows only selected or certain types of microbes to grow. Both types, general purpose and specific or specialized types of media, are very important in laboratory settings. Oftentimes we start out with a general purpose media when we're working with an unknown microbe because it provides a lot of different nutrients and allows us to grow the widest possible number of microbes in that situation. And then once we have enough of a microbe and we've started to kind of narrow down what we think it might be, we might provide it with a specific or specialized medium to further our studies. They both can come in liquid and solid forms, so you can have general purpose medium that comes in liquid or general purpose medium that comes in solid. You can also have specific or specialized types of media that comes in liquid or solid as well. You will get a chance in lab to work with different types of both general purpose and specific or specialized medias. So media provide the space and the nutrients that are needed for microbes to grow. And when we get microbes growing in media, that is called having a culture. We have a culture of microbes. So a culture basically is just the growth of microbes in or on a medium. And when we talk about cultures, there are a couple of different important vocabulary terms. Two are pure versus mixed. A pure culture has just one type of microbe in it. And a mixed culture is one that is composed of multiple types of microbes, so two or more microbes. So if you have a culture of E. coli cells, and that is the only type of microbe that's in there, just E. coli, that is a pure culture. If, though, you have a culture that is made of E. coli and Staph aureus cells, that would be a mixed culture. Sometimes we can at least get a good idea of whether we're working with a pure or mixed culture by visualizing them. So on the left-hand side here, this is a picture of a pure culture. You can't always tell for absolutely sure just by looking at it, but you can get a good idea if you look at the color of the growths on the medium. You look at the size of what we call the colonies. And a colony, I'll stick with blue ink here. A colony is a single group of microbes descended from one or a few cells. So I'll circle some colonies here. This is a colony, this is a colony, this is a colony, this is one. And basically all the individual little dots you see on this medium, these are colonies. So when we're looking at a culture and we're trying to decide whether it's pure or mixed, some of the things we do is look at the color of the colonies, the shape of the colonies, the elevation, the margins of the colonies. And as long as all of that is uniform, we're pretty certain we have a pure culture. If, on the other hand, we have a plate that looks like this, so we have our media inside what's called a Petri plate. I'll write that out. You guys probably have seen that in lab already, but just in case not. That's Petri, P-E-T-R-I. So if we have a bunch of different types of cultures, different colors, different sizes, they're not continuous, they have different margins, etc., we can tell we're working with a mixed culture, which means we have two or more different types of microbes in here. So in this culture, we have at least, let me see what color would be good to use here, maybe black. So we've got this type, this type, this type, this type, this type, and these types. We've got at least 6 different types of microbes in here, probably more than that. So basically this is just giving you some vocabulary terms and some general understanding of what we're looking for when we are growing microbes in a laboratory setting. Once we have enough microbe growing on or in a medium to actually be able to study it and to do some analysis with it, oftentimes one of the first things we do, especially if we're going to use a microscope, is to do staining. So staining is a process where we take some kind of dye or some kind of colored agent and we apply it to a sample of a microbial culture to give that culture and those microbes in that culture some color. Like I mentioned on one of the previous slides, most microbes are kind of clear in color. Not all of them, but most of them are. And if you were just to take some microbes from a culture, from some type of medium, and put them on a slide and look at it under a microscope, chances are pretty good you wouldn't see much because the light's going to pass through them, there's going to be no contrast really, and you're not going to see anything. So for us to be able to see microbes, we need to provide some color or some contrast. And we do that through the use of dyes. There are lots of different stains that can be used. You guys will get lots of practice doing what's called a gram stain in lab this semester. You will also do some simple staining. You may do some negative staining. You will look at stains called capsule stains and flagellar stains and all different kinds of stains, endospore stains. None of these are ones that you need to know right now for this particular slide, which is why I'm not writing them down. But basically, there are lots of different stains out there that allow us to view different types of microbes, as well as view different structures that microbes might make. In general, we can divide stains into two very, very broad categories. There are simple stains, and then there are more complex stains. So simple stains tend to use just one dye. And in this process, they just give any microbe in that culture that we're working with the same color. So this is an example of simple staining, just using one dye. All of the microbes, and in fact all of the cells in that culture, are just the same color. This culture happens to be taken from the human mouth, so we've got some human skin cells in there and some microbes in there as well. So what we're looking at is a situation where even though we have dramatically different cell types, human cells and microbial cells, they're all the same color because we've just used one type of stain and given them all the same color. Now, another type of stain is called a differential stain. So a differential stain is one where we use multiple dyes, typically. And the purpose of a differential stain is to allow us to distinguish one microbe from another, or a part of a microbe from another part of a microbe, or a microbe from a human cell, etc. So differential stains allow us to tell the difference between different types of microbes or different types of cells or different structures on cells. Most of the time, differential stains use multiple dyes. And as an example, you're looking at one here in the upper right-hand corner. This is an example of what we call a gram stain. And like I said, you guys are going to get a chance to practice your gram stain quite a bit in lab. And we can see there are two different colors here. There's a dark purple color, and then there's a light pink. The reason for that is because the different microbes that are in this culture have different characteristics. So they have a difference in their physical traits. And the reason that we have the different colors is because the purple dye attaches to certain structures, the pink dye attaches to other structures, and because the ones that are colored purple have structure A, and the ones that are colored pink have structure B, we see this difference in the color here. So that's why you see the difference between the different colors in a differential stain, is just the organisms or the cell types you're working with have different characteristics. Now, once we get things stained, we generally want to put them under a microscope so that we can start to see the microbes that we have grown and provided some color to. So microscopes allow for magnification, which means making things bigger, and differentiation, which means being able to tell the difference between one organism and another organism. There are lots of different types of microscopes for lots of different types of situations in microbiology labs. There are two very broad categories, and those are the light microscopes and electron microscopes. You guys will be using light microscopes in laboratory settings. The reason for that is that light microscopes can be fairly small and compact, and they don't take a lot of training to use. Electron microscopes, on the other hand, are very large and they take a lot of training to use. Now, within each of these categories, light microscopes and electron microscopes, there are different subcategories. So the specific type of light microscope you guys will be using in lab is called the bright field microscope. This is one of the most common microscopes to use. And the images that you're looking at up here on the top of the slide, those were created with the bright field microscope. So the way a bright field microscope basically works is you have a light source that shines up through the specimen, and then you have a series of lenses that you look through, and you can see the image of the microbes as a result. We also have things like fluorescent microscopes that rely on fluorescent light. We have things called dark field microscopes and confocal microscopes. There's lots of different types of light microscopes. Again, these are not ones that you're necessarily going to have to know for an exam or anything, which is why I'm not writing them out on the slide. But generally what I want you to know is that there are lots of different microscopes, and we use them depending on the different situation that we might be working in. The most common is going to be the bright field microscope. That's what you guys will be using. But depending on where you go in your career, what you end up doing, you may use different types of microscopes, or at least become aware of different types of microscopy, depending on the samples that someone needs to work with. Electron microscopes are used to look at very, very small things. Light microscopes are used to look at things like bacteria, fungi, protozoa, helminths, stuff like that. Electron microscopes are used to look at things like viruses and anything smaller than a virus. So single proteins, or sometimes they can get so good you can see individual molecules. But basically, electron microscopes are a very specialized type of microscope used to look at things that are very, very small, or to get extreme detail on something that's just a bit larger. And they take a lot of training to use. They take up a lot of space, and they're not going to be used as commonly in healthcare fields, at least in the types of settings that you guys will be working in, as things like the different types of light microscopes. Once we have grown our microbe in some type of medium, and we have maybe provided it with some contrast by giving it some type of dye, we maybe looked at it under a microscope, we might have a pretty good idea of what the microbe is, but we might need to do some additional testing to figure out exactly which type of microbe it is. So to be very specific in terms of identifying exactly what type of microbe we are working with in a setting, oftentimes we use something called biochemical or metabolic testing, or we do some kind of genetic analysis where we take the sequence of the DNA or the RNA of the organism. Biochemical or metabolic testing relies on the fact that individual types of microbes use nutrients differently. And because they use nutrients differently, they produce different types of waste products. And because of that, by using nutrients differently and producing different types of waste products, we can evaluate what microbe we are looking at by looking at what types of nutrients it consumes and what types of waste products it produces as a result. So in metabolic testing, basically what we do is we use different types of media, all of them used in the biochemical or metabolic testing typically fall under the specific or specialized medium. So we use different types of medium, and we put the microbe that we want to find the identity of in these different types of mediums. We look at what types of results we get, and then we can compare that to known standards to figure out what type of microbe we're using. So sometimes this media can come in tubes. You guys are going to get a chance to actually work on identifying microbes by using media that looks like this right here. And then other times that media that we might test our microbe with comes in these little vial-shaped containers or little oval-shaped containers like this. So if we want to do kind of certain types of testing, we can use this type of test strip right here. Other times we might use a set of media in tubes like this. Sometimes we'll use different petri dishes or petri plates full of different types of media to identify our microbe as well. And then our last way of identifying microbes is through DNA or RNA sequences. This is the most exact method of identifying microbes. So it is the most precise, most exact method. By using this method, we can pretty much always, basically 99.999% of the time, guarantee we've identified the microbe correctly. Using something like biochemical or metabolic testing, we can get a pretty good idea most of the time, but every once in a while there are some errors in that. But with something like DNA or RNA sequencing, we are almost guaranteed to get an exact result. And what we do with DNA and RNA sequencing is basically extract the nucleic acid that we want, whether that's DNA or RNA. We make a lot of copies of that nucleic acid, and then we analyze that nucleic acid to figure out the order of the A's, the G's, the C's, and the T's. Because every organism on Earth has its own unique sequence of A's, G's, C's, and T's, we can identify microbes based on this type of genetic testing. We can also identify people and plants and animals based on that type of genetic testing as well. So it's a similar method in terms of identifying a person and identifying a microbe looking at the genetic material. But this method is, while it's very precise, very exact, it does take a little bit more time oftentimes than something like biochemical testing. It can take a little bit more skill and a lot more training to be able to do correctly and make sure there are not any errors in it. So it's not used as often as something like biochemical testing or maybe things like staining and microscopy. However, it is used and it can be used when we need some type of exact identification of a microbe, either in a medical or research setting.